So, very good morning to all. So, today we will discuss about our next topic, that is importance of biochemistry and what are the properties of water, pH of water and buffer. First of all, before proceeding for the importance of biochemistry, we should know what is biochemistry. As the name suggests, it is chemistry plus biology. See, basically, we obtained energy by eating food and by drinking water, because both food and water are important to carry out basic metabolism in our body. And how do we get energy? We get energy by eating food, and that food which we eat contains certain biomacromolecules, which includes protein, carbohydrates, vitamins, and fats. So these are the four kind of major molecules: proteins, carbohydrates, vitamins, and fats, which are very important for survival and to conduct out various metabolism reaction in our body. And along with that, without water we cannot survive. So water is very important part in our life. It also basically carries certain chemical reactions, biochemical reactions, which are used in the process of respiration and breathing. Besides that, water has a different metabolic role. So in this topic today, just we will generalize discuss about what is biochemistry, what is the chemical properties of water, what do we mean by pH, and what do we mean by buffer. So we know that water is the universal liquid solvent which is found globally. Water is available in the form of rainfall. It is available in the ocean. Lot of ponds are the source of water. Lakes, rivers, glaciers, waterfalls. So about 75% of our earth is covered by water. But it is very strange that out of 75%, only I think 1 or 2% of water is available for drinking. So this is a major challenge. Preserve the water for the future use. We don't have to waste the water, because water is one of the very valuable resource which nature is providing to us. All our life based on water. So before going further, we should know what is the chemical property of water. We all know that the molecular formula of water is H2O. Okay, it contains 2 hydrogen ion and 1 oxygen ion. And water is called as polar solvent. Polar means which can dissolve in all kind of liquids, and non-polar, which cannot be dissolved in any kind of liquids. Okay, so water is having very polar nature. It is dissolved in all kind of polar solvents, but it is not dissolved in non-polar solvents. Suppose if we will add water in the oil, what will happen? The oil layer will comes out. Okay, so we will understand first little bit chemistry about the bonding of water. So the chemical formula of water is H2O. Since it contains hydrogen ion, so the chemical bond is what? Chemical bond is basically the hydrogen bond. Okay, in that, since there are two atoms, oxygen is highly electronegative. See, we can see this figure. This is the two hydrogen atom, oxygen atom. They are bonded with each other. One is having electropositive charge, one is having electronegative charge. So oxygen is highly electronegative element. Why highly electronegative element? Because it contains -2 valency. Okay, so what will happen? So it can take 2 electrons to complete its octet to come in the stable state. So if there is H2O, 2 hydrogen ion will come, they will attach on the surface of oxygen. So since it is deficient in 2 electrons, these 2 electrons will be given by 2 hydrogen atoms, and whole reaction will be complete. So this, it has hydrogen bond donor and acceptor, and water has very high boiling point, high melting point. It is very high heat of vaporization, means that around 100 degree we can say that water has very water evaporates to start. Okay, and it is surface tension. Why surface tension? Because water can assume any shape according to the vessel. Suppose you pour the water in the glass, it will take the shape of glass. Suppose you will take water in the jug, it will take the shape of the jug. Means water can adapt to any kind of shape in which it is poured. If it is poured in a test tube, it will appear test tube like structure. So all this occurs because of surface tension. So the bonding in water is hydrogen bonding. Oxygen is highly electronegative. It is having hydrogen bond donor and acceptor. It is having high boiling point, high melting point, high heat of vaporization. It has high surface tension so that it can assume any kind of shape. Okay, so that it can assume any kind of shape. Okay, we can come to the next slide. Here we can see that what is the actual mechanism of hydrogen bonding of water. See, the total distance hydrogen bond is of 0.8 mm. Since we know that oxygen is having electronegative and hydrogen is electropositive. So the distance between the hydrogen bond is 0.8 nanometer, and overall distance between the electrovalent hydrogen and oxygen is 0.28 nanometer. So the overall bonding distance, the overall chemical bonding distance between these two elements is how much? 0.28 nanometer. Now we can see that water exists in three forms: solid, liquid, and gas. We know that solid form is ice. All of you have used ice in the home. So this actually this is the see what happened exactly when this figure shows that one hydrogen molecule can associate with another 4 hydrogen molecule. Okay, when one water molecule can easily associate with 4 hydrogen molecule, it will form tetrahydral structure. It is a kind of tetrahydral structure, and we can see that it is a crystal lattice structure. So when water molecules appear, this type of compactly packed structure, they will appear in the solid form. So what they will form? They will form ice. So this compact lattice structure is called ice. So we can say that in the ice there are 4 hydrogen bonds per water molecule. Okay, so there are 4 hydrogen bonds molecules per water molecule. But when the phase of ice is converted into water, means we will provide some heat, and during heat what will happen? Ice, that is solid form, will convert into the liquid form. So it will change into the water. When it will change into the water, the number of hydrogen bonds will decrease. So now they are having about 2 to 3 hydrogen bonds per water molecule. Okay, so they are having how much? 2 to 3 hydrogen bonds per water molecule. So this is the crystal structure of ice, and it is the liquid form of ice. Right? So the overall bonding distance is 0.28 nanometer, and one water molecule can associate with 4 other water molecules. They can form tetrahedral structure, and there are 4 hydrogen bonds per water molecules in ice, and there are about 2 to 3 hydrogen bonds per water molecule in the ice. So this generally forms the compact lattice structure of water molecules, which we call as ice, that is the solid form of water. Okay, so this was the general overview about the different chemical bonding and chemical structure of water molecule. As I told you, it contains highly electronegative oxygen. So the valency is complete by 2 hydrogen atoms. That's why the molecular formula is H2O. So these 2 hydrogen ions will go over the oxygen ions, and they will complete the valency. So the bonding is called as hydrogen bonding because it involves the exchange of hydrogen ions. Then next point, we discuss about pH. Okay, basically it is the pH which shows about the acidity and alkalinity of any kind of solution. Okay, so pH basically, first we know about the history. The pH was discovered by Sorensen in 1902. Okay, so the pH was discovered by Sorensen in 1902. The pH is basically, it can range from 1 to 14. Like if pH is 0, 1, 2, 3, 4, 5, 6 till 7, it means the solution is acidic. If the pH is 7, it means the solution is neutral. If the pH is more than 7, the solution is alkaline. But the exact pH of water, though we call it 7, but it is not the exactly pH. It is around 6.8. The exact pH cannot be never 7. So the pH of water is slightly less than 7, that is 6.8. So we can say that water is very slightly acidic in nature. So it is calculated as the negative log of hydrogen ion. Suppose the hydrogen ion concentration is 1 into 10 to the power minus 7. And I told you that it is calculated as the negative log of hydrogen ion. So the pH is 7. Whatever the concentration is negative, that is of hydrogen ion, that is the pH of any solution. So we can say using the log scale, the pH is calculated as the negative log of hydrogen ion. So the neutral pH is 7. If it is more than 7, it is alkaline. If it is less than 7, then it is acidic. If it is less than 7, then it is acidic. So this is the overall about the pH. So discovered by Sorensen in 1902. It is calculated by the negative log of hydrogen ion. If negative log means whatever the concentration is of the hydrogen ion in the nature, that is the exact pH. Suppose the pH is, we can say 10 to the power minus 7, then the pH is 7. If the solution is 10 to the power minus 6, it means the pH is 6 and the solution is acidic. So based on presence of hydrogen ion, we can assume whether the solution is acidic or it is basic in nature, right? Then we discuss about buffers. Buffer is one of the most important solution which is having very prime importance. Okay, so first of all, we should know what are buffers. See, buffers are basically just aqueous systems that resist changes in the pH when small amounts of strong acid or base are added. Suppose we are using one chemical reaction. It is either acidic or basic. Suppose the solution is acidic. We want to increase the pH of the solution. What we will do? We will add base. So what will happen if suddenly we will add strong acid and the strong base? It can cause toxic reactions. So for that, we have to add buffer so that they can maintain the change in the pH. I will give you very good example. In our body, lot of biochemical reactions are there. We have protein, carbohydrate, fats, vitamins. So in that, we have a very important component in our body that is called as blood. We know that blood is having RBC, WBC, platelets. So blood is having the important role of buffer. Okay, blood is having important role of buffer. It can control all kind of pH. So it can resist all kind of changes in the pH in our body. Right? The buffers are the aqueous system that resist changes in the pH when small amount of strong acid or base are added. So we have to take very carefully when we add strong acid or strong base. We have to make buffer solution. For example, we can say sodium carbonate, sodium bicarbonate. Okay. So there are a lot of buffer solution in our body. So we can say the blood in our body mainly, the blood is acting as a buffer because it resists all kind of changes in the pH which are occurring in our body. Otherwise, what will happen? If we have strong acid in our body, our intestine will burn, we will die. If we have strong base in our body, we cannot digest the food, right? So the blood is the important component in our body which can control the resistance in the change in the pH. So basically the buffer solution contains what? Weak acid and a strong base, or just opposite, a strong acid and weak base. Okay, it can contain both weak acid and strong base and strong acid and weak base. Means out of acid or base, one thing should be strong and one thing should be weak. The most effective buffer occurs at the region of minimum slope on the titration curve. So experimentally, if we can calculate exactly the concentration of buffer solution based on certain experiments. We will not discuss this experiment because it is not in the scope of the current topic. So we should know. So they are calculated around pKa. What is pKa? Buffers are effective at the pH that are within plus or minus pH unit of pKa. pKa is the basically we call as dissociation or association constant. Suppose we are taking strong base, suppose we are taking NaOH and we do the reaction with weak acid, that is acetic acid. So NaOH plus CH3COOH. What will happen? Water molecule will evaporate. What will remaining? CH3COONA. What will get? Sodium acetate, CH3COONA. So that CH3COONA is a kind of buffer solution which is made by weak acid and strong base. So that sodium acetate we can use in our different chemical reactions. Okay, that is a kind of buffer solution which resists the changes in the pH. So in our body, I give the example about blood. That is very important buffer solution. Okay, so the buffer solution is very having of prime importance in our living system. Is that clear? So this is the example of buffer solution. Then we discuss about some compounds, that is about 4 major macromolecules, that is carbohydrates, proteins, fats, vitamins. These are the 4 major macrobiomolecules which are abundantly found in our body, and without that the individual cannot survive. The prime importance of protein, carbohydrate, lipids and vitamins. Besides that, all other are minute macromolecules. So today we will discuss about each and important macromolecules in nature. Okay. So first of all, we will discuss about first group, that is carbohydrate. Okay. We will discuss about in detail chemically and biochemically. So we should know first about carbohydrates. What are carbohydrates? They are most abundant class of biomolecules. Means they are found in largest quantity and largest diversity. They are very important macronutrient. They are very important macronutrient. They perform various physiological functions in the body. Now, what are the various physiological functions in the body? Respiration, Krebs cycle, glycolysis, respiratory system, circulatory system, all kind of biological system, circulatory, nervous, excretory, integumentary, respiration, glycolysis, Krebs cycle, all kind of physiological functions are performed by the carbohydrates in the body. Besides that, sometimes they can cause certain disease in our body. If excess quantity of carbohydrates we will consume, they will cause certain disease. Very prominent example is diabetes mellitus and lactose intolerance. Diabetes mellitus, we know, it is one of the largest occurring disease throughout the world. It is causing largest death, in fact more than cancer. Okay, most of the patients, they are infected with the diabetes, the largest infected disease in the world. No doubt scientists are trying to design any drug which can control the diabetes. Various herbal formulations are available in the market to control diabetes, but till date it has not been eradicated. So it is a major problem to control the diabetes mellitus in living beings. Another disease is lactose intolerance. Sometimes what happens, just there is a birth of small baby, and that small baby feed on breast milk for the mother. So when the baby feed the breast milk of the mother, it cannot digest the lactose which is obtained from the milk of the mother. So as a result, what will happen? The small baby will die because it cannot digest the milk which he is taking or she is taking from his or her mother, which is feeding. Okay, so these are the 2 major pathological diseases which are caused by excessive intake of carbohydrates: diabetes mellitus and lactose intolerance. So it is basically they occur due to the deficiency of insulin. Okay, because the pancreas are not able to synthesize insulin, which generally cause diabetes disease. Okay, so the detailed mechanism is beyond the scope of our topic. So just we should know that excessive intake of carbohydrates cause 2 disease, that is diabetes mellitus and lactose intolerance. Then we discuss about the chemical definition of carbohydrates. Okay, see, it contains 2 words, carbo and hydrates. So C for carbo, H for hydrates. It means definitely they will contain carbon and hydrogen. Okay, what they are made up of? Carbo and hydrates. So we will divide towards carbo and hydrates. First letter of carbo is C and first letter of hydrates is H. It means they contains which elements? Carbon and hydrogen and oxygen. COH. These two are the major elements which are present in the carbohydrates. So basically they are having the functional group which is called as hydroxyl group. Okay, so they are having polyhydroxyl group, means they are having more than one hydroxyl group, and that hydroxyl group it is denoted by OH. Okay, that hydroxyl group is denoted by OH. And then we have the functional group. What is the functional group? It is a specific group of atoms or bonds which are part of the larger hydrocarbon chain. What are functional groups? They are the specific group of atoms or bonds which are part of larger hydrocarbon chains, and they provide the specific chemical behavior. So suppose we are talking about acid. In the acid, we are having functional group COH because it is specific for its chemical behavior. Suppose we discuss about phenol. Phenol is having what? Hydroxyl group, that is OH. So the functional group of phenol is OH. Suppose we discuss about aldehyde. Suppose acetaldehyde, that is CH3CHO. So the functional group of aldehyde is CHO. So based on the functional group, we can determine the specific chemical behavior of the compound, whether it belongs to which group. Either it belongs to acid, aldehyde, for example NH2. NH2 belongs to amine. Suppose because we study about CONH2, it belongs to amides. Based on the function group, we can easily differentiate the various biochemical compounds. Okay, so the basic elements which are present in the carbohydrates are what? Carbon, hydrogen, oxygen, and they are having more than one hydroxyl group, that is OH. And based on the functional group, we can easily determine the chemical behavior of the compound. Either it can be phenol, it can be amine, it can be amide, it can be acid, it can be aldehyde. Based on that, we can easily identify. Then, as I discussed, for carbohydrates, basically there are two functional groups are present. That is aldehyde group or ketone group. As I told you, for carbohydrates, the functional group is the carbonyl group. The carbonyl group contains what? Carbon and oxygen. If it contains carbon, hydrogen and oxygen, it is having what? Aldehyde group. If it is having keto group, it is having CO group. Okay, if they are having keto group, they are having CO group. Okay, so they are having basically two types of functional groups, that is aldehyde or ketone. What they are having aldehyde or they are having ketone. Aldehyde is CHO group, ketone is CO group. Is that clear? Aldehyde is HCO group and ketone is CO group. Right? Then comes chemical structure of carbohydrates. Very clear that they contain three elements: carbon, hydrogen, oxygen, and they have two types of functional groups, that is aldehyde, which is having CHO, and ketones, that is CO. Now, next point we discuss about how we can classify the carbohydrates. Okay, so basically carbohydrates are classified based on four different their groups. Okay, carbohydrates are basically classified into four different groups. Like the unit is monosaccharides, disaccharides, oligosaccharides and polysaccharides. Saccharides are the functional unit of carbohydrates. Mono means one. If it is one unit of repeating chain, it is called monosaccharide. If there are two units of repeating chain, they are called as disaccharides. If they are having 2 to 6 unit of repeating chains, they are called as oligosaccharides. If it is more than 6, repeating chains, they are called as polysaccharides. So the classification of carbohydrates, basically they are classified on the basis of 4 different function groups, that is monosaccharides, disaccharides, oligosaccharides, as well as polysaccharides. Right? Now we will discuss about each class in detail. Okay, now we will discuss about each class in detail, that how we can easily differentiate between these different classes of carbohydrates. Okay, so first group name is what? Monosaccharides. What I told you, these monosaccharides are those carbohydrates which cannot be hydrolyzed further into the simple carbohydrates, because what I told you, they are the most simplest repeating units which is having only one chain of repeating carbon. Okay, so what I told in monosaccharides, they are those carbohydrates which cannot be hydrolyzed further into the more simple carbohydrates. Okay, it means that they cannot be further hydrolyzed into the simple carbohydrates. Thus, they are the simplest form of carbohydrates. Okay, thus they are the simplest form of carbohydrates. Okay, what are the examples? Common example: glucose, fructose, ribose, galactose. We have suppose we need energy. Suppose we are running the race and we are tired. So we get energy from what? Glucose solution. You have seen many athletes taking the solution of protein and glucose because they will provide the energy. And fructose, it is the sugar which we eat. Fructose basically is the kind of most sweetest sugar. Then ribose and galactose. So they are the familiar examples of monosaccharides, that is glucose, fructose, ribose and galactose. So these are the most simple units of carbohydrates. We cannot be further classified into simpler ones. So they are categorized as the simplest form of carbohydrates. So that was about monosaccharides. Then further we can divide monosaccharides on the basis of their functional groups. As I told you that carbohydrates contains 2 types of functional groups, aldehyde and ketone. So based on further functional groups, they are again classified in 2 types, aldehyde or ketone. If they are having aldehyde groups, they are called as aldoses. Means if the monosaccharides, they are containing aldehyde group, that is CHO, we call as aldolase. Means A for aldehyde, A for aldolase. And if they are contain ketone group, that is CO group, they are called as ketosis. They are called as ketosis if they are containing the ketone group. They are called as ketosis. Then depending upon the length of carbon chain, they are also further classified. If there is 3 carbon chain, we call triose. Tetrose means 4, pento means 5, hexo means 6, heptose means 7. So easily, suppose on the basis of function group and carbon chain, we can easily identify the monosaccharide. Suppose I talk about trioaldose. So which function group it is having? Aldose, that is aldehyde. And I told you about what? Triose, so it is having 3 carbon chain. Suppose we talk about ketohexose. Keto means which group? Ketone group. And hexose means 6 carbon chain. So easily, based on the function group and on the carbon chain length, we can easily classify the monosaccharides. So it is not the difficult task to classify them. Okay, then we can see this figure. See, we will see this figure. It is how many chain? 1, 2, 3. See, 1, 2, 3, 4, 5, 6. So this is the 6 carbon chain. It is having oxygen and hydroxyl group are there. So at the second position, this group is there, aldehyde. In this figure, which group is there? Ketone. So this group, which is having 6 carbon chain, an aldehyde group, it is called as glucose. And this group, which is having ketone group, it is called as fructose. Carbon chains are same. See, CH2 is same. 5th carbon position is same. 4th carbon position is same. 3rd carbon position is same. The difference lies where? First and second position. Because it is having the aldehyde group, so it is called as glucose. So if there is a 6 carbon chain and it is having aldehyde group, what we will call it as? We will call as glucose. And suppose we are having 6 carbon chain and we are having which group? Ketone group. We will call as what? Fructose. So fructose is the hexo keto monosaccharide. Glucose is hexo aldo monosaccharide. So easily we can identify based on the functional group and based on the number of carbon chain that it is having which kind of molecules, which kind of monosaccharides. Then we will see some examples of monosaccharide. See, first example is glyceraldehyde. It is what? 3 carbon chain. Because it is having which group? Aldehyde group, so it comes under aldo. It comes like in another example, if 3 carbon chain is there, it is having ketone, what we would call dihydroxyacetone. Because it is having which group? Keto group is there, that is CO group. Similarly, ethros is 4 carbon chain molecule and having which group? Aldehyde group. This is what? Ethylose means 4 carbon chain having which group? Having keto group. So we called as ethylose. So what we call ethylose. Another time, 5 carbon chain pentose. If it is having aldehyde group, we called as ribose and xylose. If they are having keto group, what we call ribulose and xylulose. So if somebody asks you what is the chemical behavior of xylulose, what we will tell that xylulose is basically 5 carbon chain pentose compound and it is having keto group. 6 carbon chain, I told you very good example, glucose, galactose, mannose, and if 6 carbon chain is there, keto group is there, it is fructose. So very simple. Very easily we can identify based on the carbon chain and based on the functional group that which molecule is there. Okay. Suppose in the exam I will ask, I can ask you, tell me the chemical nature of ethros. What we will get to know? Ethros belong to which group? First of all, aldehyde group, and it is having the chain of 4 carbon chain. So it is a kind of ethros. Right? So very easily we can identify the monosaccharide. It is not the difficult task. What is it we should know? We should know the number of carbon chain. We should know the functional group, whether it is aldehyde or ketone. Then we discuss about a straight chain structure of typical monosaccharide. I told you it contains how many chain? 6 carbons chain. We are talking about glucose. See, 1, 2, 3, 4, 5, 6. It is having how many carbon? 6 carbon. So the last carbon is called as terminal alcohol carbon. Why? Because it contains which group? OH group, and it is 6th position. So last thing is always called as terminal. Suppose there is 20-20 match and somebody is throwing 20th over, what we will say? Terminal over of the match. Just before last terminal, it comes penultimate carbon. If we talk about 19th over, it is what? Penultimate over. So penultimate carbon is carbon number 5. Penultimate carbon is carbon number 5. Then first carbon on which it is having CH group. First carbon is called as anomeric carbon. Okay, we should know the difference position. First carbon is called as anomeric carbon, and carbon number 2, 3, 4 they are called as epimeric carbon. Okay, carbon number 2, 3, 4 means those carbon which are connected with the first functional group of the carbon. They are called as epimeric carbon. So what we discuss, first carbon is always called as anomeric carbon. Last carbon is called as terminal alcohol carbon because it contains OH group. Okay. Second last carbon is called as penultimate carbon just before the fixed carbon. First carbon is called as anomeric carbon. And then carbon number 2, 3, 4 they are called as epimeric carbon. So based on the position of the carbon we should call them. If I talk about carbon number 1, what we will call anomeric carbon. If we talk about carbon number 4, what we will call epimeric carbon. If we talk about terminal carbon, what we call carbon number 6, that is terminal carbon. If we talk about penultimate carbon, it is what? Carbon number 5. These are the different ranging of carbons. Now, suppose what happened in this slide, we will discuss about the rotation of the carbon molecule. We know we have two hands, right and left. Similarly, based on the rotation of the carbon, we can assign them dextrorotatory, means D tape, means it is right side orientation. If the molecule is rotating left hand side, it is lever rotatory. If the molecule is rotating toward the right hand side, we call it as dextrorotatory, D. D is for right hand side orientation. If the compound or the molecule is rotating left hand side, we call it lever rotatory, it means left hand side. But who will decide? Which group of carbon will decide the rotation of the molecule? It will depend upon the penultimate carbon. Okay, it will depend upon the penultimate carbon. See, compare these two figures. Penultimate carbon is this, which is blue color. Which is blue color, that is the penultimate carbon I told you. See, in the right hand side figure, the position of hydroxyl group is on the right hand side. See, the position of hydroxyl group is on the right hand side. So we call it as what? D sugar, dextrorotatory sugar. But see this molecule here, the position of OH group is on which side? It is on left hand side. Here the position of OH group is on which direction? It is on left hand side. So we call it as what? Levorotatory or L sugar. So I meant to say that basically it depends on the penultimate carbon whether it will rotate right hand side, that is dextrorotatory or D, or whether it will rotate left hand side or levorotatory. So penultimate carbon position is very, very important. Based on that, we can say compound or the molecule is D or it is L. Right? So very simple concepts are there. Concept should be easily clarified. Now see, we will discuss another term that is called as isomerism. We all know that compound which are chemically same structure but structurally they are different. What are isomers? Those compounds which are chemically same but structurally different. It means to say that they are mirror images of each other. Suppose we are combing our hands in the mirror. What will happen? All size, our shape, the size of our face, direction of our face is changed. It means we are mirror images of each other. Those compounds which are functionally exactly the same, but structurally they are different. Why structurally they are different? Because they are mirror images of each other. What are those? They are what? They are mirror images of each other. So they will show D and L isomerism. See, here also it is showing 6 carbon chain. Here also it is showing 6 carbon chain. See penultimate carbon, carbon number 5. This yellow colored OH is on the right direction, so we called as D-glucose. In the next figure, the yellow color OH is on which side? Left hand side. Otherwise, whole chemical compound is same, but the position of OH is same, that is on right hand side. The position of OH is on the left hand side. So we called as what? Dextrorotatory or levorotatory. So based on the position of the penultimate carbon or the functional group, we can easily classify them as D-glucose or L-glucose. Okay, then we will discuss about another concept. So what are isomers? Isomers are those compounds which are chemically same, but structurally they are different because of the position of the functional group on the penultimate carbon. Then we reach to another group of compounds. Another concept is called as epimerism. So epimerism is different from isomerism. See, isomers which are different from each other only just because of the difference of the OH group on the asymmetric carbon atom are called as epimers. So those isomers which differ from each other only based on the position of hydroxyl group, their naming is changed, they will call as epimers. See this diagram. First group is CHO, second is HOH, third is HOH. But see the fourth position. In the fourth position, we have changed the direction of the OH group and H group. In this figure, OH is on the right hand side. In this figure, OH is on the left hand side. And then fifth carbon also same. And last terminal carbon also same. So here the position of OH group is changed. At which carbon? At carbon number 4. Here the position is changed. Here the position is changed on the carbon number 4. So what we will call as D-glucose and D-galactose. So we can say that D-glucose and D-galactose, they are epimers of each other on carbon number 4. We can say that both D-glucose and D-galactose are epimers of each other on carbon number 4. Clear? So easily we can identify that 1, 2, 3, 5, 6 all are same. But at the fourth position, the direction of OH is changed. So this one, when OH is right hand side, called as D-glucose, and when OH is on left hand side, we call as D-galactose. So biochemically, we can say that D-glucose and D-galactose, they are functional epimer of each other with respect to carbon number 4, because at carbon number 4 the position of HOH and OH has been changed, right? So we talk about isomers. Isomers basically depend on the position of the penultimate carbon, and epimers depend on the position of the OH group on the asymmetric carbon. Asymmetric carbon means those carbons which are in the between, either not terminal, not penultimate, not anomeric, in the between. Okay, then we talk about monosaccharide derivatives of biological importance. So what are their importance of monosaccharide in the nature? So see, this monosaccharide helps in oxidation and reduction products. When they go for oxidation, like when aldehyde will go for oxidation, it will produce acids. So it will produce sugar acids. And when aldehyde will reduce, it will produce what? It will produce sugar alcohols. They will produce amino sugars. They will produce sugar phosphates. Okay, they will produce amino sugars. They will produce sugar phosphates. So these are the main derivatives of biological importance: sugar acids and sugar alcohols. Okay, so if there is oxidation in monosaccharides, they will produce acids. If there is reduction, they will produce alcohols. Besides that, they produce amino sugars and sugar phosphates, which are of prime importance. Then we discuss about another concept, that is Mathur rotation. What is Mathur rotation? It is a term given to the change in the compound in the optical rotation position. Okay. When the compound is rotated in the optical rotation, it is called as Mathur rotation. The optical rotation is done basically at certain angles. Okay, for example, glucose, it can be obtained in two forms, either alpha or beta. Glucose can be obtained in two forms, either alpha or beta. The two forms is having different physical properties, because here what we have done, we have changed the optical rotation of the molecule, because optical rotation has been changed. Okay, so what will happen? They will change their physical properties, and they will have different melting point and optical rotation. So because of this change in the optical rotation at certain angles, the structure both physically and chemically of compounds are changed, and that phenomena is called as Mathur rotation. When either dissolved in water, because their position is changed when they are dissolved in the water. Eventually, when there is an optical rotation of the compound, so both solutions is having the same rotation, whether alpha or beta. No doubt, their rotation is almost same, but their properties will little bit differentiate. So Mathur rotation is a change in the compound based on the optical rotation, and which can change their physical properties like melting point and optical rotation. Okay. Then see this diagram. In this diagram, all positions are changed. It is exactly the same, but one is alpha D-glucopyranose, another is beta D-glucopyranose. HOHO, HOHO, HH, HH, H CH2H2, H CH2H. Here O, here H. So what we have done here, we have transferred this oxygen molecule from this position to this position. In the alpha form and the beta form, what is the difference? Difference is just only in the optical rotation of this molecule. And this, here it is rotated, come to this side. So this angle is around 18.7 degree, and alpha glucopyranose will rotate around 102.2 degree. Okay, so alpha D-glucopyranose will come to 102.2 degree, and beta D-glucopyranose will come to 18.7 degree. So this is called as Mathur rotation. So finally, after being dissolved in water, both alpha and beta forms slowly change their position. Alpha will be around 36% and beta will be around 64%. Alpha will be around 36%, beta will be around 64%. So what we discuss in today's topic, we discuss about importance of water, bonding in water, we discuss about carbohydrates, types of carbohydrates, types of monosaccharide, disaccharide, polysaccharide. We discuss about the chain of carbon, triose, pentose, hexose. We discuss about functional groups in the monosaccharides, aldehyde, ketones. We discuss about isomers. We discuss about epimers, change in the position of hydroxyl group, alpha D, dextrorotatory, levorotatory. Then we discuss about epimers. Epimers change in the hydroxyl group at the asymmetric carbon. Then we change about penultimate carbon. Then change in optical rotation is called as Mathur rotation. It exists in two forms, alpha and beta. So they are both equilibrium stage when there is a formation of about 36% in the alpha form and 64% in the beta form. So this was the topic which we discussed about today lecture. Just brief introduction of carbohydrates and water. Thank you very much.